So we're going to be talking about interactivity with the R spatial ecosystem and specifically focusing on gene expression and interpolation through kriging. So I'm just going to start by going over the technology that we're going to be using, well, the spatial technology that we're going to be using, and that's Visium. So again, we've heard about this in a number of different sections, but the kind of low resolution nature of this kind of lends itself quite well to gene expression and interpolation. So as we've said before, we place tissues on specialized slides, and these slides have these spots. And within these spots are the primers that binds to cDNA that contains spatial barcodes that we can sequence and relate back to specific spots and relate those to the tissue. And so with these spots, they're 55 micrometers in diameter, so they're quite low resolution. They contain anywhere between 1 to 100 cells, and they also have 100 micrometers distance between center to center distance between them. And that means that in between these spots, there's a whole lot of dead space that's not being captured, or it's not capturing transcripts from these regions. And so what we're going to be aiming to do in this tutorial is to kind of get around that slightly using gene expression interpolation. So we're going to use a process called kriging. And so this is traditionally used in like geographical science and all, things like that, where they would take sparse measurements, such as our Visium data, such as these points here, and then try to calculate what the likely value for a particular variable is between those. So that could be like height of the terrain or mineral deposits, but in our case, it's going to be gene expression. So if we say take like this spot and this spot, we can then kind of calculate in between what is the likely value going to be. And so we're going to apply this to gene expression data. And then, as I also mentioned, the Visium spots are very low resolution, so they contain anywhere between 1 to 100 cells underneath them. And so we also kind of want to take that gene interpolation data and map that to single cells. So one of the processes with Visium is that we take high resolution images of the tissue, and these are stained with hematoxylin and eosin. So hematoxylin is used to stain the nuclei. So we're able to identify individual nuclei and we can then map that back to our gene expression data through interpolation. So for this, we're going to be using the mouse brain data set that Jocelyn used in day 1 and day 2. So if you want to use the output you got from that, feel free to, but we're going to just be creating a quick Giotto object using this data set. One thing to also note is that we need to generate a GeoJSON file that contains a nuclear location. So we're not going to be doing that today, but we've provided it for you, sorry. But we have, and we're not going to teach you how to create that ourselves, but we have links to tutorials. So if you do want to apply this to your data sets, please click into the links here to learn how to do that. So we're going to begin by downloading the data sets. So if you haven't done that already, please do so. These are the exact same ones that we used on day 1, and we're going to extract these files. We've also provided some as a Zenodo link here for the segmentation, so the GeoJSON files, as well as other files you need for this section. And we've also supplied a subcellular Giotto object that we have created in advance that we'll be using later in the tutorial. So we're going to first begin, I'm going to jump to code now. So we're first going to begin by reading in this data. So I would recommend, if you haven't downloaded the Zenodo link, please get onto that because that might take a little while. We're going to download the data, and we're just going to start by creating a Giotto object. So I'm going to go through this again quite quickly, as these are very much the same steps that Jocelyn used on day 1. We're going to start by defining directories as well as instructions. So that's the show plot, save plot, and where we want to save the directory to. We're then going to read the data in. So that's defining where we've put it, gene index column, and the instructions. We then want to remove any spots that are not in our tissue sections, and then we want to filter based on gene expression. So again, normalizing at statistics. So for running the interpolation, we do only need to get up to this PCA step here. We're going to run through the rest of the clustering steps in order to visualize the differences between our interpolated data versus the raw Visium data. But like if you're wanting to just run interpolated data, we just need to get up to this PCA step. And so from here, we have a clustered mouse brain data set that we're going to use for the analysis going forward. So I'm just going to run this and I'm going to go over it. This will take a little while. So the first thing we need to do before we create, well, after creating our object, is to identify spatially organized features. So we're going to use BinSpect to do this. And so we're going to use this to identify genes that are spatially organized within the tissue. And the reason for this is when we create the data, this is going to give us the nicest looking results. So you can, in theory, perform feature interpolation on any gene that you want. But if we take, say, a gene that's expressed in every single cell, it's just going to be attributed to every single nuclei within the tissue. So we're not getting any further information out of it. So once this finishes, we're going to be performing the gene interpolation with about 15 genes. So I would normally recommend that you do more than this. So you want to be doing at least 500 or more genes. The reason we're only doing 15 for this tutorial is it will take too long to run some of the later steps. So we will be providing a data set that has 1,500 genes that have been interpolated, and we'll be using that for the final stages of this tutorial. But in order to learn how to perform gene interpolation, we're going to be just using 15. So this is taking a little while. So if anyone has any questions or they're a bit confused, feel free to jump in and ask something whilst we wait on this. So we're basically done here. So now we're just going to take the top 15 features that we got out of the BinSpect result and use these for interpolation. So if we just take a quick look at our object prior to interpolation, we can see all the normal things. So we have cell RNA, normalized scaled data, spatial network, PCA UMAP, and all of that. And so we run, we're going to first run this future plan function. So this is just going to allow us to perform the interpolation using multiple cores. So if you don't want to run multiple cores, please feel free to comment this line out or just don't run it. I'm going to run this. So this will take a little while to show any progress. So if you're running on a single core, it should update basically automatically. But when running multi-core, it will take a little while and then it should just kind of zoom right through. Whilst we wait, I can show you what the output of this is. Oh no, we cannot. But what we are looking for is we're going to be outputting into, if it was going to update. So now we can see it's updating it was updating the bar, and we've outputted a total of 15 images. And so these look like, not that, where did it go? Raster. So these look like this. So we get a visual representation of the gene expression in a raster form. So this is for PANTR1. And if we take a look at our object, we can see that there is now 16 images. So each one of these images here has been attached to our Giotto object and can be used for interpolation. So we can see an example of what one of these images looks like here. So it's basically we're just adding in images that contain gene expression data. So anything that's in, if I make this bigger, anything that's in white shows gene expression. Anything that's in black is no gene expression. And so now Now that we've done that, we want to map these onto the nuclei. So the first thing that we need to do is to prepare our nuclei data. So we're going to be using the segmentations from the Zenodo download. So we're going to first of all read these in using the create Giotto polygons from GeoJSON file. So for this, we need to tell it whether to load it in, we need to give it a name, and whether or not to calculate centroids. And these are loaded in a flipped orientation, so we also need to flip these. So we plot our normal Visium data. We can see kind of low resolution, more of an outline of the brain. But if we show our individual nuclei data, we can start seeing many more structures within the brain. Boom, boom, boom. So again, I just want to bring our attention to our object at the moment, and we only have cell polygons. So once we add these using the add Giotto polygons function, where we supply the Giotto object as well as the polygons that we want to add, we can then see that we now have under polygons started cells. So we have a new polygon in the exact same Giotto object as before, and these are both stored separately to each other, but within the same object. And so we can also take a look at poly info. So using the get polygon info, so Giotto will assign all of these polygons its own unique poly ID so that we can identify them. So using these polygons, we now want to calculate the overlap between the images that we have here and the polygons. So we're going to use the calculate overlap polygon images function. So this takes a Giotto object, the name of the overlap, so in this case it's RNA, the spatial information, so that's the polygon that we want to use in that starter cell, as well as the features that we want to be performing the overlap for. So these are the names, going to be the names of the images from here. So again, where are we at? If I could, yeah, that's cool. So this one's going to be more of a normal scale bar, time bar, so we can see it going across. So the more genes you want to perform the overlap for, the longer this is going to take. So currently this only works in a single threaded environment. So I think for the 1500, that took us somewhere between like 1 to 2 hours or more to run. So it really takes a lot longer when you run more than a handful of genes, which is why we're only doing the 15 for today. And so after this one is just calculating the overlap within the polygons. This next function, overlap to matrix, will be creating an expression matrix for each one of these genes that we're interpolating. So that means that for each nuclei that we provide, we're going to have expression data for every single cell compared to just the Visium cells that we had before. And the second function doesn't take. Oh, wow, that ran the same thing again. All right, sorry about that. So the second function shouldn't take anywhere near as long to finish. So now, if we take a look at our object, oops, never mind, not that. If we take a look at our gene expression data, we can see that each one of these polygons has gene expression data for all of the genes that we provided. So now we're going to kind of change gears and we're going to load in the a different Giotto object. So this is going to be the one that has 1500 genes that have been interpolated compared to the 15 that we did above. So we're going to use this for further analyses. So there's no differences other than the number of genes that's been interpolated. So we take a look, we can see that we have 1501 images. So that's the 1500 genes plus the image that we supply when we're loading in the Visium data set. So from here, we've generated basically just a new spatial unit. So we can perform all of the same analyses that we performed previously. So I'm just going to go ahead and start these because these will take a little while. So again, bear in mind that the bigger your data set, the longer that these will take. So we're going to expect that these will take a little bit longer than they'll take a little bit longer than the normal Visium data. And so if we take a look at some of our gene expression, we can see where it's localized in, say, the normal Visium. So at the moment, this is just our regular old Visium data. And then if we run the Krieg data, we can start to see the individual nuclei. And I'll show you. So one for PNTR1, which should nicely localize to these structures here. And then if we look at our Krieg data, we can see the localization. See if I can make that maybe a little bit bigger. This might take a little while. If it wants to zoom in, no. There we go. Sometimes these will, no, not letting me zoom in further, but we can see it nicely localizing to the structure within the brain. And so from here, we can also run our normal analyses. So we can run the PCA, and we're going to run through everything up to the end of clustering on this data set. So again, if these will take a little while, so if anyone has any issues, please chime in and just let us know, and we can go over those. Or if you have any questions you want to ask, feel free to jump on the microphone and just ask. So what I should have, whilst we're waiting, what I should have mentioned is that we're now telling it to use a different SPAT unit. So I think as other people have gone over before, we can change the active SPAT unit. So we don't need to specify this, but throughout we've specified what this spatial unit is that we're wanting to use, which in this case is the starter cell, as this is what we named the individual nuclei. So all of this information will be stored separately to our normal cells that we have from Visium. So all of this will just be stored within the Giotto object. And the other thing to probably keep in mind is that compared to, like, say, your normal Visium data, you will need to change some of the variables, such as like nearest neighbor, minimum distance, and like the number of clusters and stuff, due to the differences in like cell location and number. So basically you would need to just play around with these in order to get a similar result to your Visium data. Like, please don't use the exact same values, as it won't give good results.
I just want to add something now that we're all waiting. I think creaking is pretty cool, and we show what it can do. The reality is the fact that we can easily integrate our Giotto Suite framework with the long-standing R spatial data science ecosystems that has been around for like decades. There's actually a lot more you can easily do right now with these like new technologies. And I just in that link, you can actually go there. You can actually see what other types of like spatial data analysis, much more complex than what we're showing today, are possible on, like, for example, species analysis and that sort of thing. And all of that can be applied on spatial omics, right? It's in the data itself is very similar to any type of geology data or species data or whatever other modeling you're trying to do.
Thank you very much, Ruben. Nearly at the end of this. So again, like the more kind of cells you do, and so the more complex your tissue, the longer you'll expect some of these to run. And especially if you start doing this on multiple data sets in a grouped format. Cool. So at the end, we get the same thing as before. We're able to make a UMAP from our from this new interpolated data, and then we can also visualize that on the data. So this is our old Visium mouse brain, and we can look at our newly interpolated gene expression seen here. So we're starting to able to see like finer structures within the data set. And we just want to get, if we want to get a closer view, we can use the subset Giotto locks function, where we can just tell it the positions that we want to subset, as well as which SPAT units. So it's important here to use all, especially if you're wanting to keep both the Visium normal cells and the Stardust cells. So if you had just put cell here, then it would remove all of the Stardust cells, and so you'd be left with just the Visium objects. And so we again visualize the normal Visium data. You can see the structures within it to a degree, but if we look at the interpolated gene expression, we can see much finer details within each of these structures. which is showing kind of the power of this method to better understand what's actually happening in the tissue, especially when we're using some of these low resolution spatial technologies. Cool. So that's all I have to show for today. So if anyone has any questions, please feel free to jump on. Otherwise, I'll hand it over to Jocelyn.